So this lecture is going to be about the eye and the retina. So we're going to dive into vision and what comprises vision, what are the components of that, kind of the initial stages for this lecture. So kind of some basic things here, what the differences in human eye and a camera. Well, a camera, you're talking about taking a still image, you just get that reflection. Retina actually works very similar. You can kind of get that projection of an image onto the back of the retina, and then the rest of your brain interprets what that image really is. The camera, you get the image straight away. The brain requires the image to be processed, but it takes the information in from the retina, which kind of acts as kind of that receptor surface of similar to a camera would. So again, we have retinas, which are going to contain photoreceptors, which are going to convert light energy into neural activity. They're going to detect differences in intensity and the wavelength of light. So that's about colors and how strong that color is. And then we also have the logargonic nucleus, the LGN, which is going to be our first synaptic relay in the primary visual pathway. We'll get into that more later. This one's going to focus more on just the eye itself. That's kind of the first thing in the thalamus. That's going to be your relay out to cortex and get the information for your brain to process. So a quick reminder about some basic properties of light. Again, the light is electromagnetic radiation, which means that everything is traveling at the speed of light. Okay. Which means as frequency goes up, wavelength is going to go down. Okay, it doesn't matter too much, but just understanding that as wavelength gets bigger, you're going to get things like red light, as wavelength gets shorter, and things like blue light, and you have the whole visible spectrum throughout. Other animals can see some of these things like UV and infrared on the outside of our visual spectrum, but again, just understand that, again, those basic properties of wavelength, frequency. Amplitude is important because that's talking about the strength of the light, but it's not going to play a role in determining like what color something is. So again, a couple other quick properties of light. We have reflection. Light can come in and bounce off a surface. We have absorption, where light will transfer to a particle or a surface. Okay. And then we have refraction, where light's going to get bent as it moves from one medium to another, which our eyes take advantage of completely to project the image of what we see in the outside world onto the back of our retina. So a couple quick things here, some really important parts. Pupil, that is the opening right here where the light enters the eye. You have the sclera, which is the whites around the eye. You have the iris, which is going to give the color to the eye. The cornea is that glossy external surface out here, which is going to protect the eye from damaging things. And then you have the optic nerve, which is going to be our main output from the eye, carrying information to the thalamus to be relayed to cortex. So if we look through an ophthalmoscope here, there's an image of the retina here, so you can see the blood vessels coming in. You see this thing called optic disc. We'll get into more of that, what that is later. And the fovea right here. Okay. We'll talk more about what those things are, but this is basically what your retina looks like. The optic disc, actually, this is the output. This is where the nerves are going to leave the eye to go to the brain. The fovea is the main central focus point. This is where your vision is very focused and very centered, so we're looking for things up close. So I'm going to take another little quick cross-sectional thing here. We can see the retina is at the very back. That fovea is kind of that central point where light projects out to. Here's that optic disc. This is that optic disc. That is your blind spot because that is where all the nerves are leaving. Okay. There actually is no retina there. Okay. You have the sclera being the protective kind of outside white part of the eye. Two things here. We have our aqueous humor and vitreous humor. Those are the two solutions. They're going to fill the outside of the eye and the inside of the eye. Kind of maintain the shape and structure. Okay. They also need to be clear so that way light can pass easily through them. You have your lens. These zonial fibers here are really important along with the ciliary muscles because those things are actually going to change the shape of the lens. We'll see why that's important here. So again, as the image comes through, it starts to bend when it hits the cornea, and then the lens will focus that light towards the back of the eye here on the fovea. Okay. Again, that's happening because of the properties of light called refraction. Now, what will happen is that lens can actually change shape via those muscles on the outside, and that's going to change the focus. So when you're looking at something that's far away, you have a flatter lens, and you can kind of see here that lens is kind of flattened out to allow for the focus to be still maintained on the back of the eye. Whereas if you're looking at something up close, that focus will be maintained on everything here. And you think about this, if you're reading stuff that's really close to you, there's other stuff around you that you can see, but you're not focused on. You're not going to get much detail with it. So all the details are going to come out on things that are really focused on the back part of the retina. And that'll change based on the shape of the lens. The pupil is also going to react to different amounts of light. So there's connections in the retina and the brainstem that control the muscles around the pupil, determine how much the aperture is going to open and close, right? It's going to continually adjust to different light levels. And I'll post the slideshow of this whole thing so you can go back and take a look at some of these links if you want. But basically, it's a reflex, right? That's going to happen on both sides of the eye, both sides of the body, reacting to light stimulation and making sure that you're not getting saturated or you're not getting too little light. Okay. Quick terminology here, visual field. That's basically how big of an area that eye can see, okay, when the eye is fixed straight ahead. So you can actually see up to about 150 degrees-ish. In terms of what your visual field is. Now your acuity, how clear you can see things with there, is completely different. But being able to see the space around you, that's about right. But then the visual acuity plays a different role. That's where you actually can distinguish information between two nearby points. And that's based on how big of a space is taken up on the back of the retina, right? The visual acuity will come into play. You're talking about things like in your periphery, right? Like when do you get acuity to decide, oh, that's not just green, it's like a green 6 or something coming in from the side. So that's what visual acuity is all about. Okay, so more structures. Here's the retina. There's a micrograph of the retina showing all the different cells labeled with some different fluorescent proteins. And what you're going to see up top, these are your rods and your cones. Okay. So those are the things that everybody thinks about. Oh yeah, those are the things that we respond to with light, right? And you're thinking, okay, that's going to be set up where the light's going to hit that, and that's going to transmit, and then you have these other cells here that are part of the retina that are going to be the things that transmit that information further along, and then you have other cells down here which are going to take that information and do more with it. Well, here's this lamina organization in cartoon form. And what you'll notice is one key aspect. The light's actually coming in on the side. So the photoreceptors are actually down here. So here's your photoreceptors. Are here, meaning that the light's actually going to make its way all the way through this entire layer until it actually hits the photoreceptors, the rods and the cones that are going to respond to the different frequencies and wavelengths of light. So the other layers that are there, the top one's called the ganglion cell layer. You can see that's connected to these axons, which are going to take the cell out. So they're connected to that optic nerve. Okay. And you have this inner plexiform layer, not too much worried about that. You have the internuclear layer, which contains these other cells, which are transmitting information back. And then you have the outer plexiform layer, which has this other cell that looks like it's going horizontally almost. But then out here you have the photoreceptor cells. So it's actually kind of inside out, since light actually has to go through the other layers first. So that means those layers have to be very thin and very clear very spread out. So that way light can actually get to those photoreceptors without being bounced around too much. So kind of break down some of the names really quick. Okay, you have the photoreceptors down here. You have the horizontal cells, which act horizontally. These photoreceptor cells are going to send information vertically back that way. And they're going to send information to these bipolar cells. So the horizontal cells will send information to a bipolar cell, like so, but the horizontal cells will modify that input. And then those bipolar cells will output onto these ganglion cells down here, which are things that are going to have the axons that project out. And here you have your horizontal input modified by these what are called starburst amacrine cells. These are starburst. And what's going to happen there is you just have that last chance to modify the output. And we'll see more about the processing and what kind of role those cells are going to play later. So again, kind of those lateral connections here. Horizontal cells are going to receive input directly from photoreceptors and project to other photoreceptors and bipolar cells. So they'll modify the input coming from those photoreceptors. The amacrine cells are going to receive input from the bipolar cells, and they're project to ganglion cells, bipolar cells, and other amacrine cells. There's lots of different neurotransmitters. There's lots of different types of amacrine cells. A lot of times they'll play an inhibitory role. They may play an excitatory role. They're going to balance out the signal that's coming out of the retina. So photoreceptors, what's going on here? So they're in charge of converting that electromagnetic radiation to neural signals. They're the initiation point for it. There are four main regions. You have the outer segment out here. You have the inner segment and the cell body, and then the synaptic terminal. Okay. The outer segment is going to be the key part for generating our signal, right? You have these little discs in here that contain the photopigment. So there's your photopigment. That's what's going to react to the light, and that comes in. And again, we have our two types that you learned about before, rods and cones. We'll talk more about later about their specificity. Now, in terms of retinal structure, where are these things? What does the structure actually look like in different locations? So as you get closer to the fovea, you have, let's see here, an extremely high concentration of cones. That blue line goes way up near the fovea, and there's basically zero cones out in the periphery. However, rods are much higher concentration in the periphery, and they drop off in that fovea. So in that central, remember, fovea is the focus point. So in your highest area of focus, you have very little rods, but you have a lot of cones. So immediately it tells you, okay, cones are used for focusing, whereas rods are used for doing kind of other things, more general things. The other thing that's going to happen too is the amount of neurons later in the process that are actually going to be dedicated to those photoreceptors. So in the central area, in that central retina, you might even have a 1-to-1 ratio, right? One photoreceptor, one bipolar cell, one ganglion cell. So it's extremely sensitive to light. Whereas you're in the periphery, you're going to get a lot of rods and cones with a lot of bipolar cells, 1-to-1 ratio there, but then you also only have one ganglion cell for a whole bunch of those. So basically telling you you're just less sensitive there to all the details of things that are happening around you. So here's a pretty cool thing about the fovea's anatomy. If you look at a cross-section of it, you can see that the layers actually have been pushed out to the side that are above the cones, so giving more direct access to the light. So it's not getting bent or going through any sort of refraction before it has access to those cones right here in the fovea. You see they're really packed in tight there. And again, that's where they're in that 1-to-1 ratio with a ganglion cell. One cone, one bipolar cell, one ganglion cell, no rods in that central fovea. And again, like telling you that central fovea, those cones, they are for high visual acuity, things you really want to focus on, things you're really trying to see, or things in the periphery like, hey, I hope we notice something's there. Right. So phototransduction in rods. So a couple things here. Light energy interacts with photopigment, produces a change in membrane potential, kind of a G protein coupled receptor. Um, the thing here though is when it gets hit with light, it actually causes a decrease in second messenger. So photopigment kind of works backwards of everything we've been seeing. So if photopigment hits it, G protein gets activated, second messenger actually gets shut down. So instead of increasing second messenger, we're actually going to decrease second messenger. And you see one of the things here that happens is actually a decrease in sodium conductance. We're actually going to get a hyperpolarization in a rod when it gets hit with light. So here in rods, you have dark. You can see that the outer segments are depolarized in the dark because of sodium's constantly coming in. However, photoreceptors are going to hyperpolarize in response to light. So you can see we're at minus 30 millivolts in the dark. Light hits, we drop all the way down to minus 60 millivolts. So basically that means we're going to stop sending information when that rod is activated. So it's kind of a reverse of what we normally see is activation happens and neurotransmitter gets released. Well, here activation happens and neurotransmitter release actually gets shut down. So it's very different. It's a very different process than what we've been seeing before. So membrane depolarizations are actually happening all the time when there's no light, and then it stops when there is light. So again, just going through the dark to light process, okay? Here you can see your disk that's sitting inside. We have our rhodopsin that's in there. That's what's responding to the light. Light hits that rhodopsin. It activates that G protein right here, and that's actually going to shut down this phosphodiesterase, and that's going to close that sodium channel. Okay, so there's our process of light actually causing an inhibition as opposed to an excitation, would kind of like the opposite of the chemical situation that we dealt with smell and taste. So in cones, similar to rod phototransduction, except now we have different options. So before we had a rhodopsin, which was just simply light yes or no, and actually extremely sensitive, just so you know, you don't need many photons of light to actually activate a rod, which is why you can see in the dark fairly well. But to activate a cone, you need much more light, and they're going to have three different cones. You're going to have blue cones, you're going to have green cones, and you have red cones. And the combination of those is what's going to give you the entire spectrum that we see through from the electromagnetic spectrum, the whole visual light spectrum. So you're going to read a thing about the evolution of color theory, and that's going to talk to you about that trichromacy aspect. How do we actually get three different colors that end up creating every single color in the spectrum? So here's kind of overlap you might see. So you have these three different ones. You have an S, an M, and an L photoreceptor. Don't worry about that too much. To understand that basically you're covering the entire range of the visual spectrum by some combination of activity levels of S, M, and L type cells. So that should hopefully kind of give a good introduction to basics of how we start to interpret the light signals that are coming in, and then we'll talk more about how that signal is parsed out for a little bit finer detail later.